In this lecture, we're going to look at the membrane protein structures, and this is really where my passion lies. We've already seen how complex biological membranes are, looking at the properties of lipids and how they assemble, and we're now going to transition to see how membrane proteins sit in the bilayer and interact based on the properties of the amino acid side chains that are present. So you should be able to describe what the essential properties are for a membrane protein, and we'll see that there are different types, really based on how they associate with the membrane. Detergents are going to be really important because they have the ability to mimic the lipids, and they'll be needed in order to remove and purify all of the membrane proteins, and specifically integral membrane proteins, while other membrane proteins called peripheral membrane proteins can be removed using milder conditions, and we'll look at some examples. And it is going to be important for you to remember all of the amino acid side chains because you can use this information to predict how a membrane protein is going to associate with the lipid bilayer. There's only one section of Stryer for this lecture, but it does have 4 subsections, but it's not too bad if you're looking for some supplemental readings. These are the different types of membrane proteins, that is, proteins that associate with the membrane, versus a soluble protein that can be found in the cytoplasm of a cell or secreted outside. Peripheral membrane proteins are proteins that interact with the hydrophilic surfaces of the membrane, so they're kind of on the edge or peripheral of the membrane. In yellow are 2 examples where A is forming non-covalent interactions with the polar head groups of lipids. B associates non-covalently with another integral membrane protein, protein C. Now, these integral membrane proteins are embedded in the hydrophobic bilayer, and these can form pores or channels and have much more complex secondary structures. Or they can just pass through the membrane once, like in protein D, and contain other parts that are either inside or outside of the cell. The third category are the lipid anchor proteins, where you can have post-translational modifications to proteins that will covalently attach a lipid tail, and that's what's going to hold the protein at the membrane. So essentially it's tethering the protein to the lipid bilayer. Let's start with the peripheral membrane proteins. Now, the operational definition of a peripheral membrane protein is something that you can remove either using high salt or by changing the pH. So remember when we talked about purifying proteins using ion exchange chromatography? We used both of these methods to disrupt the non-covalent interactions between the protein and the column. And for peripheral membrane proteins, this will either disrupt the ionic interactions or change the protonation state of the protein, releasing it from the membrane. So there could be individual amino acid side chains that are, say, positively charged, like lysine or arginine, that will interact with those negatively charged lipid head groups that we saw. And you can also see hydrogen bonding holding the protein to the membrane. Or in the second example, you can have an amphipathic helix where one face is hydrophobic and interacts with the membrane. Now there are some peripheral membrane proteins that have little hydrophobic loops that kind of dip into the membrane and then come back out. And they are different than an integral membrane protein that would be more tightly bound. The fourth way is that a protein can stay peripherally bound by interacting through weak non-covalent forces with another integral membrane protein. We've looked at some amphipathic helices before, and these generally have one side of the helix that's polar and the other side that's hydrophobic. So if you're looking down the long axis of the helices, you can see the polar ones on one side and the non-polar ones on the other side. You can also look down from one end to see where the side chains are pointing around that alpha helices. Remember, you need about 4 amino acids for a complete 360 rotation, so you'll generally see an alternating pattern of 2 hydrophilic and then 2 hydrophobic amino acids if you were to look at the primary sequence. The nonpolar groups will then interact with the hydrophobic lipids and allow it to embed in the membrane. There are a number of tools online that you can access that can allow you to generate the helical wheels that show you the different properties. An example of these types of helices would be found in prostaglandin H2 synthase that has an amphipathic helix that attaches it to the membrane, allowing for it to carry out its function. You can start to build up a helix by typing in amino acids, and what you can see is that the amino acids will start to build up the helix from a top-down view, where there's about a 100-degree rotation for the addition of each amino acid. And you can see that the different properties of the amino acids are color-coded, with the hydrophobic amino acids in yellow on one side of the helices and polar red, green, and blue ones on the other side. Now, you can have the odd amino acid that isn't hydrophobic, such as this green threonine or serine, but in general, the majority will be hydrophobic on one face. And the same goes for the opposite polar face of the helix. You could have the odd hydrophobic amino acid present. The one thing to keep in mind is that you can't really continue indefinitely with this 2 hydrophobic then 2 hydrophilic pattern, since it does start to rotate around and there are only 3.6 amino acids per complete turn. These computational tools allow you to assess the properties of a sequence of amino acids, and you could go on and enter your own polypeptide, say one representing your name in one-letter codes, and see what kind of a helix could be generated. Integral membrane proteins are more tightly associated and span the lipid bilayer, and we generally refer to the polypeptide chains that are going through the membrane as transmembrane segments, or TMs. Because they do span the membrane, they do require harsher conditions like detergents or organic solvents. Detergents kind of look like the lipids and can replace the lipids and coat the membrane protein in order to solubilize them for purification. So these detergents are also going to be solubilizing the peripheral proteins, but if you can't get a membrane protein out of the lipid bilayer using high salt or a change in pH, chances are that it will be a membrane protein that's an integral membrane protein like these green and purple ones shown in this figure. We'll look at the different structures that they can have, and remember, some of these may also be glycosylated on the extracellular surface. So these branch structures that are covalently attached to some of the amino acid side chains. And you can also have multi-subunit membrane proteins that associate in the membrane as well. We can classify this group of membrane proteins based on how the polypeptide chain travels across the membrane. Remember, we always write proteins from the N-terminus or the NH3 group through to the C-terminus. And if the N-terminus is found on the outside and the protein only goes across the membrane once or has a single TM, this would be a type 1 integral membrane protein. Type 2 would be flipped, where the amino terminus is on the inside and the C terminus is outside. And a type 3 integral membrane protein would have multiple TMs. And for these proteins, you can either have the N-terminus on the inside, as shown here, or on the outside. Where the C-terminus lies really depends on how many TMs are present. So an even number would result in it being on the same side as the N-terminus, and the multiple TMs would then be connected by loops that are generally hydrophilic in nature. Type 4 are multipass as well, but they contain multiple polypeptides that come together and associate to form a complex in the membrane. The lipid-anchored proteins also fall under other categories, and these form stable interactions with the membranes that will also need harsh conditions in order to remove them. Now, these lipid tails are going to be covalently attached to the functional groups in the protein, so either the N-terminus as shown here, or maybe some of the side chains. Type 5 membrane proteins would have a single lipid anchor, so this example here would be an N-myristylation. So the lipid is attached to a nitrogen group, that's what N stands for. Or you could have type 6 membrane proteins that have transmembrane regions as well as that lipid group attaching and anchoring it to the membrane. So in this case, it would be S-palmitoylation, where we're covalently attaching to that sulfur in the side chain. The final group are GPI anchored proteins. So if you remember from the last lecture, lipids can have sugars attached to them, and GPI stands for glycosyl phosphatidylinositol. So these membrane proteins are attached to a phosphatidylinositol lipid, so this would be found in the membrane, and then that lipid has a sugar chain attached to it, which would then be covalently attached to your protein. And this would be a form of post-translational modification. As I've mentioned before, membrane proteins are a bit trickier to work with. We do need to use detergents to help solubilize them, while milder conditions could be used to disrupt some of the more weakly associated peripheral membrane proteins. You could also use organic solvents, but these may not preserve the structure of the membrane protein, allowing you to study its structure. These detergents essentially create a shell wrapping around the membrane protein and replacing the hydrophobic lipid tails. This creates what's called a micelle, and provided the detergent is mild enough, the protein doesn't really know that it's left the membrane, and you can preserve its structure for purification and analysis. We've already seen an example of a detergent called SDS, but this is quite a bit harsher in nature and can denature your protein. This table just shows you some of the structures of some commonly used detergents. You can see that they are amphipathic in nature, where there are hydrophobic regions like these chains, and those are the regions that will interact with the hydrophobic portions of your membrane protein. The hydrophilic portions are what helps keep your protein in solution so that you can study it. Now, as I mentioned, some detergents are strong like SDS, but there are other ones like Digitonin or Triton X100 that are a bit milder, and these will allow membrane proteins to keep their folded structure. We describe detergents based on their critical micelle concentration, where at this concentration, they will start to form these micelles where the hydrophobic regions can come together. And when you're above the critical micelle concentration, or the CMC, you start to see them forming around membrane proteins. Now, you don't need to memorize these numbers, but should know what the critical micelle concentration means if you were to use it in the lab. So it'll be important to calculate how much you're going to add into your buffer, in addition to the buffering molecules that will help keep your protein happily folded if you are indeed trying to study its structure. Because membrane proteins are more difficult to purify and study, another aspect that's really important is trying to figure out the topology. So if you don't have a structure, how can we figure out what the organization of the polypeptide is across the membrane? Now, we've already looked at protein sequencing, and you can potentially predict the protein structure based on a DNA sequence. So perhaps it's some novel membrane protein that no one's studied before. And if you know the primary sequence, one thing we can do is examine the amino acids present and see if they can form alpha helices that are hydrophobic enough to span the membrane. Now, there are membrane proteins that have beta barrels in them, but when we talk about TM or transmembrane segments, they're usually alpha helices. If you think about the structure of an alpha helix, the hydrogen bonds run down the middle and are shielded, and there's a rise of about 1.5 angstroms per amino acid. So based on the typical width of a membrane, you would probably need about 20 hydrophobic amino acids to span the membrane. So we've already talked about how to classify amino acids, but do need to be a little bit more precise. And we can use the primary sequence to predict whether or not a protein is found in the membrane. If you were to find long stretches of hydrophobic amino acids in the primary sequence that are maybe separated by some polar amino acids, this could correspond to the transmembrane regions that are buried in the membrane, and then any hydrophilic loops that would be on the surface connecting different TMs. We've already seen this table in the amino acid properties lecture, and we do generally express an amino acid's polarity in terms of its hydropathy or hydrophobicity, and that's the relative degree to which an amino acid is hydrophobic. Keep in mind that amino acid hydrophobicity is not an absolute term, it's a relative term expressed as a number. And then we can rank all of the amino acids on scales called amino acid hydrophobicity scales. So these scales are developed based on different calculations and experimental observations. So things like their solvation energy and solubility, as well as their partition coefficients, and by observing where you typically find them in purified proteins. We're going to focus on the Kite-Doolittle scale and look at two ways you can arrive at these types of numbers. These scales originally started popping up 40 years ago to give amino acids a score or number to describe how hydrophobic they are. Nowadays, there are also many computer algorithms that can be used to predict their properties and membrane protein structure. Now, there are two main techniques that are used in order to arrive at this number, either taking an experimental or a visual approach. The first uses organic chemistry to look at how an amino acid partitions between two immiscible liquids. So you may have used one of these separation funnels in your chemistry labs, and what you would do is include an aqueous and an organic phase that essentially don't mix. You would then add your amino acid, give it a really good shake, and allow them to separate into the two layers like you see in this image. If you were to use octanol, which would mimic the lipid interior or a very hydrophobic environment, you would expect hydrophobic amino acids to separate and be found in this sample. You could then measure the concentration of your amino acid in each of the two phases and calculate the fractional distribution. The second approach was done by Chothia in the 1970s, and this involves averaging out where the amino acids are found in known protein structures. At the time, only 12 protein structures were used, but you could now do it using the thousands of structures that we have to get an even better estimate. Essentially, what you'd be doing is looking to see how buried each residue is. So on the right is a structure of carboxypeptidase A, which we've talked about before. And if we just use lysine as an example, so we've got these blue residues on the surface, these charged amino acids, and say we have 10 lysine residues in total, where 8 of them can be seen on the surface, that means 2 of them are going to be 95% buried. To determine the hydropathy score, we would just divide 2 by 10 to give you 0.2. If we do it for isoleucine in orange, we can see that there's 2 at the surface, but if there were 10 total, you would have 8 that are at least 95% buried. So 8 divided by 10 would be 0.8. So it's a combination of evidence that can be used to arrive at this scale and give each amino acid an identity. Some of these numbers you kind of expect, like the amino acids that only have hydrocarbon tails, so isoleucine, valine, and leucine are very hydrophobic, while things like proline actually comes out as being a bit more polar. And this is simply due to its presence in beta turns, which can be found at the surface of proteins. Now, someone was asking about cysteine at the beginning of the course, and it does come in as being more nonpolar. And that's because it can be found in these disulfide bonds, or cysteine, and those would be buried inside of a protein. We can then use the Kite-Doolittle hydrophobicity values to figure out if a stretch of amino acids is hydrophobic enough to span the membrane. You could just look at the value for every amino acid in a sequence and see if you can find 20 that are very hydrophobic. But remember, you could have the odd one that is hydrophilic, so we use what's called a sliding window or averaging approach. This involves determining the hydropathy index for a stretch of amino acids. What you would do is add up the values for each amino acid in a segment of known length, then divide by the number of amino acids used to calculate this average hydropathy index. You would then slide the window that you used for the segment, where the window is how many amino acids you are averaging. You'd slide it one residue along the chain and then recalculate the average hydropathy index. We can then plot the results where you would use the central amino acid in your window as the X value and then plot the hydropathy index on the Y-axis. Now you can do this calculation using a different window size, so say 7 amino acids long, or you could do it for 20 amino acids, which is how many span the membrane. But remember, you are averaging out the amino acids on either side of that central amino acid. So there's really only going to be one value that gives you that super hydrophobic score. Once you move the window, you're starting to incorporate the polar residues outside of the membrane. So we do generally use smaller windows, either 7, 9, or 11 amino acids. And these can generate noisier plots, so plots with higher peaks and valleys, but it's these peaks that we're looking for, very high hydrophobicity scores that span at least 20 amino acids. When you use a larger window, say 20 amino acids, you would see smoother plots, but they will have shallow peaks and valleys. And if the peak isn't high enough, it might be hard to determine if a TM is present. Let's just look at a sample calculation. You would write out the hydrophobic score for each amino acid from the Kite-Doolittle hydropathy scale. You'll then choose a window of, say, 7 amino acids, and then add up each value and divide by 7. You would then assign that number to the central amino acid, in this case threonine, and that would be the number that you would plot on the y-axis, where your x-axis number would be 4. We then move the window one position to the right, and recalculate the average hydropathy index and plot it for the fifth amino acid, leucine. These are two extensively studied membrane proteins and the hydropathy plots you would get if you were to work through their primary sequences. Glycophorin A, or GPA, is a type 1 membrane protein that has a single transmembrane segment. And you can see this in the graph when you work out the average hydropathy indices and plot them relative to the amino acid residue. There's a single large positive peak that spans at least 20 amino acid residues, but this doesn't necessarily tell you where the TM starts and ends. You would need to examine the primary sequence in more detail to figure out exactly which amino acids may be crossing the bilayer. But this is one way you can start to elucidate the structure of integral membrane proteins. GPA is also heavily glycosylated, so you can see many sugar chains on the outside, and these contribute to different blood group antigens. Bacteriorhodopsin, on the other hand, has 7 TMs, and these are numbered on the hydropathy plot. And its function is to move protons across the membrane, and this occurs when it undergoes a light-induced conformational change. Here are some links to different hydropathy programs where you can enter a sequence and it'll generate a plot. The last one was actually developed by Dr. Dieber, a member of the Department of Biochemistry, and your assignment will actually get you to look at membrane protein structure and generate one of these plots. So this lecture really is just a brief overview, and we do have upper year courses like BCH 422 that will look at membrane proteins in more detail, but you should be able to appreciate the importance of non-covalent interactions for the attachment of these proteins to the membrane. We can separate and purify them using different techniques where increasing salt and pH can remove peripheral membrane proteins, while integral membrane proteins require detergents. We can also start to predict the structure of a membrane protein by analyzing the primary sequence, and we can use the hydropathy index for each of the 20 amino acids to generate these hydropathy plots. Make sure you do go back and understand how to generate and read these plots before doing the brain break questions.